Welcome back viewers to Dr. Raktim Mukherjee's biology channel. Here we make biology concepts simplified. Today's morning talk or video is an extension to our earlier videos on apoptosis. We have observed the video on extrinsic pathway, the intrinsic pathway, role of caspases. Further continuation, we shall delve into details the terms and terminologies associated with apoptosis. In detail, we shall study apoptosis, necrosis and necroptosis. What are the points? What are the differences? How exactly they can be experimentally studied? So when you talk about the mechanisms of apoptosis, necrosis and necroptosis, that shall feature in today's video. Mind you students, this talk I am going with cell biology point of view. So first I shall brief diagrammatically. If you see, this is the normal cell, and then if you see the left side, this is N, necrosis, and the right side is A, apoptosis. Let's look at the diagram very critically. If you look at the diagram critically, you can find this particular apoptotic body. The presence of apoptotic body is a hallmark sign of programmed cell death or apoptosis. Later on, the same apoptotic body is subjected to phagocytosis. In other words, phagocytes do clear all the apoptotic bodies, making the cellular process rebound back to normalcy. Look at the left side of the panel. This is necrosis. In necrosis, there is enzymatic digestion and leakage of cellular components. You can see there is a leakage of cellular components. Now, if I use an analogous situation, I always say apoptosis is like a person who is committing silent suicide. That person only knows that the person is dead. He won't disturb even to the nearest person of his or her life. Whereas in necrosis, it is more like show off. The person will indulge in screaming, shouting. In other words, this is analogous to, in necrosis, one cell will damage the neighboring cell. In apoptosis, one cell will have no effect on the neighboring cell. So these are the highlights using diagram. We move further. First, we will study apoptosis and necrosis based on morphological features, the same morphological features that we studied just now in diagram. The very first point in necrosis is, in necrosis, the cell loses the membrane integrity. When I say the cell loses the membrane integrity, normally a healthy cell will have a intact cell membrane. So this cell membrane integrity is lost, complete loss is there. Whereas in case of apoptosis, there is not complete loss of membrane integrity, but there is membrane blebbing, but no loss of integrity. So someone asks you, in which form of cell death you find membrane blebbing, it will be apoptosis and not necrosis. Second point, I'll number it 1. This is second point. The second point, this begins with the swelling of cytoplasm and mitochondria. I'll show it diagrammatically. This is the normal cytoplasm C. When necrosis sets in, the cytoplasm will swell, it will increase in size, that is swelled cytoplasm. Likewise, the mitochondria was of normal size. Because of swelling, the mitochondria will also increase in size. So this begins with swelling of cytoplasm and mitochondria. Apoptosis, here it begins with shrinking of cytoplasm. I will show you what is shrinking. Right, this is the normal cytoplasm. When I say shrinking, it will reduce its size, it will constrict, that is shrinking of cytoplasm, and condensation of nucleus. The nucleus will become more condensed under microscope. Morphological means you can observe under microscope. Further, in necrosis, it ends with total cell lysis. Complete cell is broken down. Total cell lysis, no vesicle formation, that is complete lysis. In apoptosis, it ends with fragmentation, not complete lysis but fragmentation. Small fragments are formed, that is smaller bodies. Finally, the last morphological point for necrosis. In necrosis, it is disintegration or swelling of organelles. I already shown with diagram the swelling. Whereas in mitochondria, the mitochondria becomes leaky. I would like to state the mitochondrial permeability complex, the mitochondria will become more permeable, MOMPS last time we studied, and this becomes leaky. Once the mitochondria becomes leaky, through the mitochondria, the proteins of, there will be a pore formation. I will draw. This is mitochondria. Once it becomes leaky, this type of pore is formed, and through this pore, the proteins, the pore formation will involve the proteins such as that of BCL2 family. So these are morphological features. Now we will study the biochemical features. So the answer is divided into morphological features, the biochemical features, and physiological significance. So now biochemical features. First biochemical feature, in necrosis, there is loss or regulation of ion homeostasis. Whereas in apoptosis, the ion homeostasis is not affected, it is tightly regulated process. Necrosis does not require energy, no energy. Whereas in apoptosis, it requires energy. Random digestion of DNA, that is smearing of DNA after agarose gel electrophoresis, you will find very classical smearing of DNA. Whereas in apoptosis, non-random mono or oligonucleosomal length fragmentation. In other words, you observe the DNA ladders in case of apoptosis. Further, post-lytic DNA fragmentation, that is a late event of death. In case of apoptosis, it is a pre-lytic DNA fragmentation. So necrosis is post-lytic, apoptosis is pre-lytic DNA fragmentation. In apoptosis, there is release of various factors into cytoplasm by mitochondria. I will draw it that from mitochondria, the contents are released into cytosol. There is activation of caspase. Caspase activation we have already seen. Caspase activation we saw in the previous video, and alterations in membrane asymmetry. So these were some of the biochemical features of necrosis and apoptosis. Now we come to the physiological significance. Necrosis affects group of contagious cells. As I said, it will scream, shout, chikna, chillana, but apoptosis is just very personal. It just affects the individual cells, no effect on neighboring cells. Necrosis is evoked by non-physiological disturbance such as immune system's complement attack, lytic viruses, hypothermia, hypoxia, ischemia, metabolic poisons. Whereas apoptosis is induced by physiological stimuli, that is lack of growth factors, changes in hormonal environment. Necrosis, there is phagocytosis by macrophages, whereas in apoptosis, the phagocytosis is by adjacent cells. It can both be by adjacent cell as well as macrophages. In apoptosis, phagocytosis is very important because phagocytes will clear the debris that are formed in apoptosis. Necrosis, most important point, I would say, the fourth point. In necrosis, you have very significant inflammatory response. So inflammatory response is present in necrosis, and in apoptosis there is no inflammatory response. Student, this is the most important point. Mind you, apoptosis causes less harm or no harm at all. Why? Because after apoptosis there is no inflammatory response, whereas after necrosis significant inflammatory response is there with the concomitant release of pro-inflammatory cytokines. We move ahead. Now we study some of the important points of apoptosis and necrosis that we have already studied. Programmed cell death is apoptosis. It involves inflammation, right? Now we study the next term. What is necroptosis? That is very interesting. It's a new term, that it is necroptosis. Necroptosis is nothing but it's a form of programmed cell death that shares feature. So I will place it in this way. Necroptosis is nothing but apoptosis plus necrosis. So when you have both features of apoptosis and necrosis, it is called as necroptosis. Lots of research papers are available. It's a very important cell death pathway. So necrosis plus apoptosis is necroptosis. It's again a programmed necrosis. It's a programmed necrosis. So mind you, alone necrosis was non-programmed, but when you talk about necroptosis, it is programmed. This is initiated by signaling pathways, that is necroptosis is receptor mediated. This ends in necrosis like death. In morphological and biochemical terms, what are the features? The features would be decrease in ATP. Whenever a cell undergoes necroptosis, there is decrease in ATP. Just like necrosis, there is swelling of the cell. There is generation of ROS, reactive oxygen species. There is release of lysosomal enzymes, and finally there is rupture of plasma membrane. So the integrity of plasma membrane is lost. There is rupture of plasma membrane. Now we will study some of the tenets of necroptosis pathway. What are the features of necroptosis? We have already seen. Now necroptosis pathway. This is the diagram of necroptosis pathway. I will divide the pathway into basic few parts. The first part I will highlight as initiation. So what is the initiation? What is the trigger? Here the trigger or initiation, what is the trigger? The trigger is cellular stress. That is the first trigger. Apart from cellular stress, you also have the next trigger. Apart from cellular stress, you also have the reactive oxygen species, that is ROS, RNS, reactive nitrogen species. These are all the major triggers. Okay, and these triggers can actually activate the necroptosis pathway. As I said earlier, necroptosis is nothing but the difference between it's an amalgamation of apoptosis and necrosis. The next point of initiation is receptor activation. When you study receptor activation, in necroptosis there is activation of death receptors. Death receptor activation takes place. Which are the death receptor that gets activated? Some of the death receptor that gets activated are TNF R1, that is your tumor necrosis factor receptor 1. This is of course the common initial point. This completes the first step, that is initiation. Our next step is formation of necrosome. So first I will write the players. The two major players over here are the RIPK1 and RIPK3. If you see the diagram, that's the RIP kinase 1 and this is the RIPK3. Okay, further you can see RIPK3 in multiple sites. So what happens? Formation of necrosome, I have already highlighted in the diagram. RIPK1 and RIPK3, they stand for receptor interacting protein kinase 1. Receptor interacting, receptor R for receptor, I for interacting, P for protein, K for kinase 1. Receptor interacting protein kinase 1 and receptor interacting protein kinase 3. They are the key regulators of necroptosis. So what happens in the formation of necrosome? By using RIP1 and RIP3, a complex is formed. I will write there is a complex formation that takes place. What is this complex formation? Upon activation, RIPK1 and RIPK3 interact to form a multi-protein complex. This multi-protein complex is called as necrosome. So RIPK1, RIPK3, they will form a multi-protein complex. This multi-protein complex is called as necrosome. This is called as necrosome. Our next step is activation of MLKL. Next step is, as I said, is activation of MLKL. Whenever you talk about activation of MLKL, this involves two major steps. The first is the MLKL phosphorylation and second MLKL activation. First we will discuss MLKL phosphorylation. In the necrosome shown in the previous step, RIPK3 phosphorylates mixed lineage kinase domain like pseudokinase, that is MLKL. What does MLKL stands for? M stands for mixed, L stands for the lineage, mixed lineage kinase domain like pseudokinase, domain like pseudokinase, that is MLKL. So RIPK3 phosphorylates MLKL. If you see this, RIPK3 phosphorylates the MLKL. Once the MLKL activation, so once MLKL is phosphorylated, this MLKL will become activated, and phosphorylated MLKL will undergo conformation change. It will become activate. I will put in diagram. RIPK3 will phosphorylate MLKL. So now MLKL has become phosphorylated. Once MLKL has become phosphorylated, then it will undergo, this MLKL will undergo, it will undergo conformational change. It will undergo conformational change, and this conformational change will make MLKL active. Once MLKL becomes active, it will translocate to the nucleus. This is how it is. It will translocate to the nucleus, and after it translocates to the nucleus, it will oligomerize and disrupt the plasma membrane, making it rupture, and it will initiate the downstream pathways carried out by necroptosis. Okay. So that was a short video on apoptosis, necrosis and necroptosis.